Hi, my name is Andreas Untergasser and I'm the developer of Primer3Plus and one of the maintainer of Primer3. Today I want to highlight the interaction of Primer3Plus with the Genome Browser, which is really handy if you're working with genes. So you can select your gene and browse in the Genome Browser to the position of the gene. And now if you select the gene, you can find a link to Primer3Plus. And if you click on this, all the information are shuffled over to Primer3Plus. And we can use it in different ways. Initially, it was thought to create primers which allow you to amplify RNA. And if you're amplifying RNA, you always have the problem that DNA might be a contaminant of it, so the genomic DNA. So one trick what you can do is you put your primers over an exon-exon boundary. And if you do nothing, this is what's selected in Primer3Plus. And it will have all the exons in the sequence here. And the exon-exon boundaries are highlighted with this minus, which selects them as primer overlap positions. So if we pick primers, one of the primers will be forced on top of this exon-exon boundary. And that is really handy because this primer will not be able to bind in genomic DNA. And so the contaminants are not such a big problem with this primer set. So in the top, we can also return to the Genome Browser and see where the primers are. And what we see now here is that we have huge primers and smaller ones. The big ones are the ones which are spanning this this intron here. So the primer starts here in this region. Here there will be no primer on the RNA because the intron is gone, and it will continue here. So you see this primer span this intron and the other ones span other introns here. And the small primers which you hardly see, they are the primers which work with the exon spanning primers. Okay, so this worked well. But it's not the only one. The only thing we can do. We can load the exon-exon boundaries as targets. Then you see that we will have this exon-exon boundary selected as targets. And if we pick primers, one of them will be amplified by the primer set. So this or that one will be amplified with this primer set. And usually this is not so handy. You can do things you like on your own. So if you're loading the genome sequence, then you'll have the exons in capital letters and the introns in smaller letters. So you can select whatever you want to do and use it as a target here and have your primers forced on the position where you want to have it. So that comes in handy. Or if you're into exon sequencing, then you can select even individual exons, for example, this one. And now you see that the exon 7 is selected. Somewhere in the sequence, we'll have the capital letters. Currently, I'm not seeing it. Let's go down. Oh. It's tricky to see where actually the capital letters are, especially if you have such a huge gene. And anyway, it will be there. We can pick the primers. At least then we will see it. Here is our sequence, and here is the exon it wants to amplify using these primers. And again, we can return to the Genome Browser. And now we see that all the primers are here in one side. And here should be the exon 7. So that's the one we will probably want to amplify. Let's zoom in and see if this worked out. Here we go. 3 times. 3 times. So now we see we have here the exon, which is the exon 7. And we have the left primers on the left side of it and the right primers on the right side. So if you want to use these primers as well for sequencing, then it's not so nice if they really glue to the exon, because you would like to have another 50 base pairs, which allow you to get a better sequence in the exon. So what we can do here is we have our target. We just subtract 50 bases on this side. No, come on. Number 1 I want to do. No, it doesn't let me change it. I have a clear idea. So now, and because we want to have 100 additionally, we have to change the length by 100 to add to it. Now we pick primers, and we can have a look in the bottom. Now the target will be bigger. Where is it? Here. We see the target is only the things in capital letters. Here we have additional 50 in front and additional 50 behind it. And now we can also return to the Genome Browser. And again, we have to zoom in. And you see now the primers have some distance to the exon. And that allows us to get a good sequence if we use the same primers for amplification and for sequencing of the exon. So what you have to do if you want to sequence all the exons, you have to go one by one through all the exons and pick primers for each of them. And that you can do. You can modify the positions here in the bottom. You can start working directly on the genome sequence. So this is quite a handy tool. But one thing you have to keep in mind, you will get the same orientation here like the chromosome is. So our open reading frame here is in reverse. So we will not find an ATG and a stop codon here in the bottom. We will find it the other way around. So we will have somewhere here a sequence of CAT, yeah, because it's the reverse of the ATG, and the stop codon will be somewhere here in the top. And if it's not in reverse, then it will write here that yeah, it's in forward. Okay, I hope these links help you a lot, and it will ease the way how you can work with genes in and pick primers. Thank you for listening and watching.